Hey everybody, welcome to V40. This is the second video on Gregor Mendel. We saw in the first video on Mendel, he came up with, in numerous studies, he kept finding the same ratio, that magic 3-to-1 ratio. Why did he come up with that? What does it mean? And how did that help him come to some understanding of genetics? I'm right here. So let's go on and see how he came up with what he came up with. So Gregor had a hypothesis. He developed a hypothesis based upon that 3-to-1 inheritance pattern he observed in the F2 offspring. Remember, we're talking about the F2, so that parental generation gives rise to the F1s. The F1s give rise to the F2s. So he kept finding that pattern. And again, it wasn't just any pattern of 3-to-1. It was oops, let me try that again. It was three dominants. It was three dominants to one recessive. That's what he found with every trait he looked at. He found in the F1 generation, he found all of one type, and that's the one he referred to as dominant. Then he had the recessive, which is what reappeared in the F2 generation, and he always found that 1/4 of the offspring, roughly, were of that type. So he always found 3 dominant to 1 recessive. Okay, here it is in the 1800s. You figure out how genetics works from that information. But he did. He inferred a model of genetics that's accurate. So he has 4 concepts. So let's look at each of these. The first one is that there are alternative versions of factors. He didn't know the word gene because the word gene didn't exist back then, but alternative versions of factors account for variations in inherited characters or traits. So he said for flower color, for example, there must be 2 versions of flower color. There must be one version for purple flowers and another version for white flowers. And I'm going to make that a little p for right now, and it'll be clear later why that's true. So these alternative versions of genes are now called, in 2015, alleles. So whenever we're talking about an allele, we're talking about a version of a gene. So the gene in this case is flower color. The versions are purple and white. So this heritable factor is what he called it, but now we know these heritable factors are genes, and he concluded from his data, well, there must be 2 versions. How did he get to that point? We'll touch on that in a minute. I forgot I had this slide. But if we look at a pair of homologous chromosomes, and for right now, just pretend that we actually have the sister chromatids attached to them. If you want to think of it like this, that's great. But it shows you on one location of the chromosome, on each of the homologous chromosomes, we have the information, we have the gene for flower color. We have a purple version of the gene and a white version of the gene, and we call these alleles. So what Mendel said is that everything has 2 versions. You have one version and a second version, and we'll talk about how he's able to come up with dominant and recessive in a second. But he said there must be at least 2, well, there must be 2 versions of a gene in this case to account for the numbers he found, that 3-to-1 ratio. Okay. The second conclusion he came to was that each character in an organism inherits 2 alleles, one from each parent. So again, it kind of makes sense to us today in 2015 that you get one version from mom and one version from dad. It wasn't necessarily clear in the 1860s. So Mendel said that when you have a male and a female, each contributes a version of an allele to the offspring. So these alleles may actually be the same. That is, you may pass on a big P from one parent and a big P from the other, or you may pass on different versions. And I keep drawing these little letters, and that'll be clear here in a second why I'm doing that. But when you look at the locus, remember that previous slide, that location on the chromosomes, they could be the same versions, big P, or they could be different versions, big P and a little p. So he concluded, though, that not only are there must be two versions, but that you inherit a version from a parent. Now, this is what I kept sort of touching on before. The alleles may be the same or they may be different. What Mendel was able to conclude by looking at the F1s, when he only found one type, he said, there must be two different versions in this case, and if that's the case, then the dominant one, and that term will be clear in just a moment, the dominant one is the one that controls the appearance. So back in remember we had a purple flower and a white flower, and when we crossed these, the F1 generation had all purple flowers. Mendel said that must be because all these F1s, all of these ones in this generation, all of these must have one dominant version and one recessive version, one hidden version of the traits. So the recessive allele is the one that has no noticeable effect on the appearance if it is in the presence of a dominant one. And this will be a little easier to see in a future slide. Okay, and the fourth idea, oh, I forgot I had this slide too. There are dominant and recessive traits in humans. If you inherit, for example, let's just focus on a couple of these. Let's say that you have a form of albinism, albino. That is because that's a recessive trait. The way that someone is a recessive, or someone who has an albinism trait, is they're going to have a little a from one parent and a little a from the other parent, and that's why they're going to be an albino. Alkaptonuria is a metabolic condition, same thing. The one parent passed on the little a version and the other parent passed on the little a version. So if you have two recessive versions, you have that trait. If you have a big A and a little a, then you don't have the trait because the dominant allele controls the appearance. Now, over here is kind of the opposite scenario in some ways. Polydactyly. This is sort of an interesting condition where you may have 6 fingers or 6 toes. Yeah. Okay, polydactyly is a dominant trait. So that means if I have a big P and a little p, then you would have the condition polydactyly. If you the only way you would not have it is if someone was a little p and a little p. Because if they have one dominant allele, one dominant version, they have the trait. So of course someone who was big P would also have polydactyly. Where are these letters coming from? I'm using the A's and the P's. Usually we just use the first letter of the word we're talking about to represent the alleles. So the capital versions are always dominant, the little versions are always recessive. But notice there are some conditions, just to kind of recap, that you have only because you have two little versions, two recessive versions of that trait. And there's some versions, another disease is a form of dwarfism. If you have one dominant allele, then you have the trait. Okay? And finally, the fourth concept that Mendel was able to figure out is that these alleles segregate independently of each other to end up in different gametes. This one's a little bit maybe confusing until you think about meiosis. Think about U for some trait, and let's just represent the trait as big C and little c. Okay, if this is you in your somatic cells and you go through the production of sperm or the production of eggs, then half your gametes, whatever they are going to contain one version and the other half are going to contain the other version. Let's pretend they're sperm for a second. Okay, because they're going to segregate independently, which means they're going to separate independently of each other. You're going to have half of your traits, half of your versions of your traits are going to end up in half your gametes, and the other versions are going to end up in the other half. And you'll see when we look at a Punnett square shortly how we represent that. Let me erase. Okay. So now the simple version of Mendel. You're like, you should have started with this to begin with. I know. Okay, what Mendel said, simple version is this. There are two versions for every trait, and those versions are called alleles. Versions of the traits are separated into gametes. That is, half the versions go to one set of gametes, the other half go to the other set of gametes. Everyone ends up with two versions for each trait because you have two parents. You got a version for mom and a version for dad for each of these traits. Dominant alleles are always expressed, recessive alleles are not. They're only expressed or show up if you have two recessives at the same time. If you have a dominant and a dominant, or a dominant and a recessive, then the dominant wins out. That's the trait we see. If you only can see the trait as recessive if you have two recessive versions, and that's what Mendel was able to conclude. Now, that was pretty good stuff. That's for the 1800s. It might be easier to see what Mendel did if you look at a Punnett square. Punnett squares are really cool. Mendel didn't invent them. Actually, they're invented by Dr. Punnett. But the Punnett square is a way of seeing the crosses between two individuals in a little easier, more visual way by doing this. Okay, it's really important to see what's on these axes. Over here represents a gender. Let's just call this the female, and over this is the male, although it really doesn't matter which is which. And one way of representing a cross between this individual, which happens to be big P, little p, it's not easy to see this on this slide. So this individual here is also big P, little p. What's represented in the middle here is if this particular sperm cell hits this particular egg cell, what do you get for the combination? Since you have a big P here and a big P here, then you get a big P zygote. Okay, if it happens to be this sperm cell hits this egg cell, then it's going to be big P, little p, as you see here. Okay, same thing, big P represented here, little p represented there. We get a big P, little p version here, and then we have a little p and a little p there. Now, notice something that I think is important. You may not, but let's pretend anyway. If you look at this box, the box is broken into fours. Okay, yeah, we can see that. Okay, why are half of the gametes from this individual big P and half little p? Because Mendel said so. Because half the sperm cells produced contain one version and half contain the other. Same thing for the eggs. So if you kind of do this, the math, you see 1, 2, 3, 4 boxes here. A quarter of all the combinations of sperm cells and egg cells should be like this. A quarter of all the sperm cells and egg cells should be like this. Another quarter should be like this, and another quarter should be like that. Now you may be thinking for a second, now wait a minute, because I have over here, I have one big P combination. Actually, this one and this one are both big P, little p combinations. Okay, and you may recognize that I have one combination that is little p, little p. Okay, remember what we said about these. If you have the dominant version, you get the dominant trait. You have the dominant version, you get the dominant trait. So I really have three of them that are kind of like this, because these are all going to have the same trait, whatever the trait is. And since we're looking at flowers, we're assuming it's going to be peas and it's going to be purple. And 1/4 of these is going to be like this. That should look familiar, a 3-to-1 ratio. When Mendel analyzed his flower color, if we kind of take what Mendel found and then put it on a Punnett square, let's see if we get what we think we should get. Mendel found that if I crossed a white flower, white is only white because it has no purple. So it must be a little p, little p. This one must be big P, because it's a true breeding plant. So now if I look at the combinations, I have big P, little p, big P, little p, big P, little p, and big P, little p. So all of these should all be purple, right? Because they all have the dominant allele. Now, if I cross this flower against one just like it, that is, these are F1s. If I cross these against each other, I put purple and purple together, big P, and get this combination. Big P, little p gives me this one. Little p, big P gives me this one. And then little p gives me this one. But notice the colors. I have these three that color-wise are all going to look purple. So I got 3 purple and I have 1 white. So that explains why Mendel saw a 3-to-1 ratio when he looked at flower color. It's also why he saw the same thing when he looked at height in peas, and when he looked at seed color in peas, and flower position in peas. All of these traits are inherited in a Mendelian fashion, which means 2 versions of a gene, okay, the alleles. You inherit one from each parent. There is dominant and recessive, and if you have a dominant one, you have the dominant trait. So, as we wrap up, we can see it like this. Purple flowers and white flowers, all of the offspring are purple because they all got a big P, they all got a big P from one parent and a little p from the other. All of them are like this. When these cross against themselves, when I cross another flower like this with itself, then half, and we'll pretend that this one, for example, is here. So you have big P, little p. Pretend this one is over there. It doesn't matter which is which. But then I get my 3-to-1 ratio of purple to white. Remember, this one looks the exact same as that one because if it has a purple allele, it's purple. Now, a term I've really had a hard time not saying because it's a very common genetic term are these, homozygous and heterozygous. So now I can finally say them. A homozygous individual is one that has 2 of the same alleles. This individual has a big B and a big B. On its chromosome, let's say it got from dad, it has a big B. On the chromosome it got from its mom, it has a big B. This person is homo, the same, zygous because they have this. You can also be homozygous if you got a little b from dad and a little b from mom. That can also make you homozygous. We call these people homozygous recessive. You could have gotten a big B from dad and a little b from mom. You could have gotten a little b from dad and a big B from mom. These people are heterozygous because hetero means different. So homozygous individuals can be either dominant or recessive. Heterozygous, they have 2 versions. They have the big B, little b. So just to kind of wrap this up, I'm going to give you two more very important terms that we use a lot in genetics: phenotype and genotype. Hugely important terms. Okay, phenotype means your appearance. How do you actually look? Okay, your genes in this case are responsible for your appearance. Your genotype are the actual alleles you got to make them up. So let's consider this. If you consider the genotype of these individuals, of the ones produced in the F2 cross, you have a quarter of these, 1/4 of all of these are going to be big P, big P. 1/4 are going to be homozygous recessive, little p, little p. And then 2 of the 4 combinations are going to be big P, little p, heterozygous. So the genotype ratio is I have 1 big P to every 2 big P to every 1 little p, little p. Okay, the genotypic ratio is that. But because this one and these 2 all look purple, when I talk about the phenotypic ratio, or the appearance of it, actually I have 3 purples to 1 white for my phenotypic ratio. So genotypes refer to alleles. Phenotypes refer to appearance. It just so happens that in the case of the heterozygotes for this cross, the purples, because they have one big P and one little p, look just as purple as this one here. Okay, this is a really important video, folks. You definitely want to come back and look at these terms, walk your way through what Mendel said, and you find yourself to understand it a lot better. Final slide, I promise, is this one, which is understand what we're saying when we talk about the DNA to phenotype journey. You have here in your cells, you have the chromosomes. The chromosomes in this case have this particular gene right here. This gene actually is going to make the protein, remember, through RNA. You see the RNA right here, makes the protein. The protein folds up, and now it actually works, in this case, to hold oxygen. So the DNA here, the instructions, ultimately results in a phenotype. In this case, the appearance is the appearance of this protein hemoglobin to carry out oxygen. If we talk about the DNA in pea plants, there is an instruction set on one chromosome that ultimately results in an amino acid sequence that produces the color purple in pea plants. Again, really important video. Do go back. You probably have to go back and watch this one again so that you understand the relationship between phenotype and genotypic ratios, and that you understand what Mendel was able to do to demonstrate why 3-to-1 works, because of the way that information is passed from chromosomes ultimately to produce the protein. That's it. Know it's a lot. Might need to watch it again. Just saying. Thanks.